This is a production of Cornell University.
Hey, hi everybody. My name is Greg Vogel. I'm an assistant professor in plant breeding and genetics. It's great to have the chance to talk to you all today about a project in my research program. So just to give a quick background on what my lab does, we breed and study the genetics of tomato and eggplant. We have a few different objectives in our research and extension program. Primarily, our first and foremost, our goal is to develop varieties for the Northeast United States in particular. We also do work characterizing the genetic basis of traits of importance. I'm going to be talking about a research project aligned with that goal. Another area that we're very interested in is modeling strategies to implement new technologies in applied vegetable breeding. A major theme there is strategies for predictive breeding approaches using genome-wide DNA markers to predict phenotypes. And then finally, we generate and disseminate results on variety performance in New York for our stakeholders, including farmers, gardeners, and members of the seed industry. So today I'm going to be talking about salinity stress. Salt stress can be a challenge for tomato production in a few different environments, primarily in arid and semi-arid regions of the world, of which New York is not one, but the Central Valley of California is. And in the Central Valley, they grow about 95% of the processing tomato crop for the United States. It's about 200,000 acres of tomato. And if you look at this map of groundwater salinity, if I can figure out how to use this, in the Central Valley, you can see that there's a substantial proportion of the acreage where they deal with salinity problems in the groundwater. That area also coincides with some of the major production regions for processing tomato. California is not in a drought situation right now, but it's likely that they will return to one in the future. And with climate change and a future of unpredictable precipitation patterns, this problem is expected to get worse. Closer to home, if you visit vegetable farms in our area, this is a common sight that you'll see, some type of high tunnel for protected culture and season extension. And when you cover the plants and prevent precipitation from falling on the soil, you get reduced leaching of soluble salts that can build up and cause problems. This can be managed through cultural practices, but it's not always easy to do so. So to tackle this problem, we've been working with a wild tomato relative called Solanum sitiens. Solanum sitiens is found in the Atacama Desert of northern Chile. That's one of the driest non-polar deserts on Earth. Actually, it is the driest non-polar desert on Earth. On average, they get about half an inch of rainfall a year. If you look at this picture of a wild Solanum sitiens plant growing, you can see how desolate the landscape is, yet this tomato relative is somehow able to survive there. For many years, tomato breeders weren't able to access genetic variation from Solanum sitiens because it's not cross-compatible with tomato. However, researchers at UC Davis spent several decades, I think close to 30 years, moving genetic material from sitiens into tomato using a really complex crossing procedure with other wild species hybrids as genetic bridges, and they were able to develop a set of introgression lines, which are lines that all share the same tomato genetic background and then have different pieces of Solanum sitiens DNA. This is just a diagram of chromosome 8 of tomato, and these segments show the individual sitiens genome segments that are present in different introgression lines in this population. So we started a project to look at these lines for their salinity stress. The way we did this, after some trial and error and some exploratory research, was we grew them to maturity in the greenhouse, and we irrigated them with drip irrigation, and we used fertilizer injectors to inject not just fertilizer, but also a concentrated sodium chloride solution. In total, we evaluated 27 introgression lines and their tomato parent under fertilizer only and fertilizer plus salt conditions. We used salt treatment that corresponded to an electrical conductivity of 10 decisiemens per meter. Here's a picture of the plants growing. This is what the salt damage looks like in more sensitive genotypes. You can see the scorching on the leaves, and then blossom end rot we see on the fruit of these lines as well. This work has been led by the senior technician in my program, Howard Rice, and my PhD student, Diana Mulder. Here's a quick snapshot of the results looking at these 27 lines. So on the left, I'm showing a scatter plot where the x-axis shows their yield under control conditions. On the y-axis is their yield under salt conditions. The red dot is the tomato parent, and then each of the dots is an introgression line with a different piece of Solanum sediens DNA. You can see that none of the lines performed as well under salt conditions as they did under control conditions. You can see that the tomato parent out-yielded many of the introgression lines under control conditions, but it was out-yielded by many introgression lines under salt conditions. If you look at the yield, not in terms of its absolute yield, but in terms of the relative difference from control to salt conditions, that's what I'm showing on the bar plot on the right. This is the percent yield decline from control to salt. The red bar corresponds to the tomato parent, and here you can see that most of the introgression lines had a greater yield stability under salt compared to the tomato parent, indicating that there are probably many genes distributed across the solanosidians that all have some effect on salt tolerance. I should say that we decided to follow through with 3 of the introgression lines for further characterization. This one here showed the highest total yield stability, although a lower absolute yield potential under control conditions. And then these 2 lines showed the highest absolute yield under salt conditions. So we looked at those in greater replication. We also crossed them back to tomato to look at the introgressions in the heterozygous state, which is important in the context of breeding hybrid cultivars. And here I'm showing the tomato in red. This plot is a little different. On the y-axis is the percent yield reduction under salt. So having points lower on the y-axis indicates greater yield stability. We verified that these 3 lines had higher yield stability. We did see, however, that in the heterozygous condition, a lot of that yield stability was lost. Ultimately, we decided that this one line, 5297, which showed about a 30% yield decline under salt, it out-yielded the tomato parent under both control and salt conditions, and its yield stability was partially preserved in the heterozygous state, was a line that we were going to move forward for future research. So there's a lot of future areas that we're moving forward in this project. We're interested in how these salt-tolerant lines performed when challenged not just with sodium chloride, but with more complex salt mixtures. We're trying to move these lines to realistic field conditions to see how these greenhouse results translate to the field. We're collaborating with a few additional SIPS researchers, including Casey Brickman in horticulture and Bala Saheb here in soil and crop sciences to understand the physiological mechanisms that confer this salt tolerance. Genetically, we're trying to see if we can reduce the size of these introgressions, get rid of extra DNA from the wild parent while retaining the salt tolerance. And then we're also interested in additional traits coming from this wild relative, including freezing tolerance that we've had a few undergraduate interns work on. And we've seen that you can put the wild species in a chamber at about -6°C for 4 to 5 hours and it survives. And we're interested in if we can find those genes and characterize those in tomato as well. So with that, I'll thank the members of my lab, the people who have contributed to this project. I mentioned Howard and Diana. We've also collaborated with Neil Madsen and Nick Kazmar in horticulture on this. And this work has been funded by the California Tomato Research Institute. So happy to take questions if there's time. Yes?
You ever take a transgenic approach to this?
No, although I'm interested as a possible application of the research. The linkage drag, or the negative traits conditioned by the Solanum sediens DNA that tags along with the beneficial genes can be a major problem. If we're able to identify a candidate gene and maybe instead of actually moving that chromosomal segment into tomato, but instead editing it, I'm interested in that as a potential outcome of the research. Yes.
There is some body of literature, doctors on salinity tolerance, and I wonder if you could use that to supplement what you're doing, more of a called it like reverse genetics approach, so to look for candidate genes that way.
Yeah, that's an interesting suggestion. There's been a lot of work in tomato also, but it's a good point. Yeah, there's a lot of genes that are already known in function that we should look at. Yes.
The next slide. Your first question is about salt mixture.
Mm-hmm.
What does it mean? It means sodium chloride combining with the mineral, and why it is necessary for your next time.
Yeah. So in the realistic conditions where plants face salt stress, there's a diversity of different ions that are contributing to that salt stress. We've looked at soil test results from farms in the Central Valley where they have salinity problems, and one of the problems is no two farms have the same challenge necessarily. So the way that we're trying to just, I guess, get a quick understanding of this question of whether the tolerance in these lines is specific to sodium chloride or generalizable to overall salt stress is we're now challenging them with a salt mixture that's resembles seawater. We're using instant ocean, which people use in their saltwater aquariums. And we're challenging them with that and comparing the response to sodium chloride. Okay, yes.
I may have missed this, but the wild tomato is collected from a place that is obviously very arid, but is it also saline or sodicic there as well to begin with?
Yeah, I should have mentioned that, but they've found wild populations of this species growing in very saline soils with an electrical conductivity as high as like 50 decisiemens per meter. The threshold at which tomatoes starts to show a yield decline is like 2.5. The specific accession of this species that was used as the parent of this population, there's no information on where that was found in terms of the salinity of the soil. So there could be genetic variation within that wild species for its ability to tolerate salt. But overall, particular accessions of that species have been found growing in highly saline environments. All right, thank you, everybody.
This has been a production of Cornell University, on the web at cornell.edu.